noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
This website makes learning so easy. I found all my resources here.
Like what you share.
Stem cell classification is vital. Potency depends on complex molecular mechanisms. These mechanisms drive full cellular differentiation. Regulatory milestones now shape therapies.
Stem cell research has undergone a radical transformation. At its core, a stem cell is defined by two capacities: self-renewal and differentiation.
Through self-renewal, stem cells divide to maintain their undifferentiated state. In differentiation, they acquire specialized functions. This intricate balance facilitates development and repair. Potency is described as the gene activation potential. It begins as a continuum at fertilization and progressively narrows as cells commit to specific fates.
Totipotency represents the zenith of cellular potential. It produces all cells, including both embryonic and extraembryonic lineages.
The zygote is the only indisputably totipotent cell. It undergoes cleavage into identical cells. This state remains until the 16-cell. A totipotent cell can generate a living organism. Pluripotent stem cells can form any of the 3 germ layers. These are the endoderm, mesoderm, and ectoderm. They build every tissue in the body. This is the trilineage potential. Pluripotent cells are remarkably versatile. They can give rise to all tissues of the adult body, naturally within the blastocyst, while engineered versions include auripotent stem cells.
As development proceeds, stem cells enter states of increasing restriction. Multipotent stem cells give rise to multiple types, but only within a specific lineage. A mesenchymal stem cell can become bone, cartilage, or fat. Oligopotent cells like HSCs produce blood lineages. Unipotent cells differentiate into a single cell type. A fertilized egg is the primary source. It forms a zygote, or early morula. These totipotent cells can form any cell type. This represents the ultimate cell potency. Pluripotent stem cells are remarkable. They originate from the inner cell mass of a blastocyst, form all 3 germ layers, endoderm, mesoderm. Examples include ESCSCs and iPSCs. Multipotent stem cells can differentiate into multiple cell types within a single lineage. Examples include MSCs and NSCs, found in bone marrow, adipose tissue, and the brain.
Welcome to the potency category. These cells form few, closely related types. Examples include hetopoietic stem cells, found in bone marrow and cord blood.
Derived from the blastocyst, embryonic stem cells are pluripotent. They can proliferate indefinitely in the lab and differentiate into all somatic cell types. This makes them vital for regenerative medicine. The blastocyst, 50 cells, it is composed of 2 distinct parts, the outer trophectoderm and the inner cell mass, which forms the future fetus.
The isolation of ESCs requires careful extraction from the surrounding trophectoderm. This process typically involves surplus embryos from IVF clinics. Techniques have evolved to maximize precision. Laser biopsy allows for precise targeting. This effectively preserves cell integrity.
Immunosurgery removes the trophectoderm. Specific antibodies and complement lies the outer layer. The intact ICM is then cultured. Feeder layers promote self-renewal. The timeline of ESC discovery is punctuated by major breakthroughs in vertebrate biology. We want Fiesthrosabelsnick biology. A significant leap occurred in isolation. These provide an inexhaustible source. Scientists first identified blood-forming stem cells. This was done through pioneering research in mice. Adult stem cells exist in many mature tissues. They serve as an internal repair system. They replenish dying cells and regenerate tissue.
Adult stem cells reside in specialized niches. This microenvironment governs their fate. Injury signals activate them to regenerate tissue.
Hematopoietic stem cells, found in the bone marrow, generate over 1 trillion new blood cells every day. They divide into long-term and short-term cells, sustaining our entire blood supply.
Mesenchymal stem cells, or MSCs, are found in bone marrow and fat. They can become bone, cartilage, or muscle. Easily isolated from adipose tissue, they are key for regenerative medicine. Neural stem cells reside in the adult brain. They can differentiate into nurse astrocytes. However, their isolation is medically invasive. iPSC are a viable alternative.
Epithelial and skin stem cells reside in the basal layer. They populate hair follicles and the intestinal lining. This ensures rapid turnover of tissues subject to constant stress.
Bone marrow contains both HSCs and MSCs. They exist in a concentration of 0.01 to 0.1%. These treat leukemia, immune disorders, and repair bone. Adipose tissue is a rich source of stem cells. These are predominantly adipose-derived stem cells, with a concentration of 1 to 2%.
They are used in soft tissue repair and as an anti-inflammatory.
Dental pulp is a rich tissue source. It contains dominant dental pulp stem cells. The estimated concentration is 0.1 to 1%. They are used in dental reconstruction and neuroprotection.
Skeletal muscle is a rich tissue source. It contains satellite cells. Their concentration is highly variable. They help in muscular dystrophy and support direct injury repair. These cells show great clinical promise. Exploring the tissue source of the oral cavity. Oral MSCs are the dominant stem cell type. They show a moderate estimated concentration, great for periodontal and gingival repair.
Driven by the reprogramming paradigm, mature somatic cells are genetically reset back to a versatile, pluripotent state.
Reprogramming drives cellular reversion. OCT4 is essential for pluripotency. KELF4 helps erase somatic programs. CMYC globally enhances chromatin kinetics. An alternative set of factors, OCT4 and Lin28, achieve similar results. The process is slow, with low efficiencies, triggering a stepwise transition to full pluripotency. PSCs advance medicine. Derived from patient cells, they avoid immune rejection. They create powerful disease models. They also bypass ethical concerns.
Perinatal stem cells are collected from tissues traditionally discarded. These cells are considered younger than adult stem cells. They possess a greater capacity for expansion. Cord blood is rich in hematopoietic stem cells. These cells can rebuild a patient's blood and immune system. This process is called hematopoietic reconstruction. Wharton's jelly is rich in mesenchymal stem cells. These cells target autoimmune diseases. The placenta aids wound healing and ocular applications. Perinatal source, umbilical cord blood. Primary stem cell type, hematopoietic. Advantages: lower GVHD risk and non-invasive. Clinical usage.
Perinatal source, Wharton's jelly. Primary stem cell type, mesenchymal. Advantages: clinical usage, autoimmune disease trials, tissue engineering.
Derived from amniotic fluid, these stem cells show high plasticity and are completely non-controversial. They are used in developmental modeling and vital spinal cord injury research. Primary stem cell types, MSCs and trophoblasts. They are readily available and rich in growth factors. Clinical usage includes wound care and ocular membranes.
Stem cell identification relies on molecular markers. This primarily uses a cluster of differentiation protocol. These surface molecules define a cell's identity. This system allows for accurate isolation. Pluripotent stem cells have specific markers. Nuclear markers include OCT4, NANOG, and SOX2. Surface antigens include SEA4. Human cells lack SEA1. Adult stem cells are vital for repair. HSCs are marked by CD34 positive. MSCs express CD73 and CD90. NSCs use nestin and CD133.
Human ESCs and iPSCs are defined by specific markers. They express positive markers like SEA1 and CD45, supporting their pluripotency and cell adhesion. Hematopoietic stem cells, or HSCs, are the foundation of all blood cells, but lack markers drive unique bone marrow homing capacity. Let's examine mesenchymal stem cells, 73, 90, and 105, but negative for CD34, DR. They provide vital stromal support and immune modulation.
Neural stem cells, or NSCs, are positive for nestin and CD133, but negative for CD34 and 45. They drive glial and neuronal differentiation.
PSCs offer unlimited potential, but pose a grave risk. A teratoma is a tomb with tissues from all 3 germ layers. Residual cells can form these tumors in grafts.
To ensure clinical safety, several techniques remove residual pluripotent cells. Antibodies can deplete teratogenic cells, while small molecules selectively target them.
Strategies are needed to purify stem cells. Lectins can strip undifferentiated cells. Suicide genes, a prodrug, kills any remaining tumor-forming cells. This ensures safer, purer cell therapies.
Pluripotency carries an inherent risk. Undifferentiated cells can proliferate. Vascular sites like the kidney promote tumors. Viral vectors in iPSCs may integrate oncogenes.
Stem cell therapy transitions from research to licensed medicine. This is governed by rigorous regulatory pathways, like the RMAT designation. Enacted under the 21st Century Cures Act, RMAT designates promising therapies with nearly 300 requests by mid-2025, driving priority review and breakthrough approvals. From 2020-25, Omasurge was approved to accelerate neutrophil recovery in patients. Lyfgenia was approved as a gene therapy to treat sickle cell disease.
In December 2024, the FDA approved Ryoncil. Bemdaneprocel shows promise for Parkinson's. Phase 3 trials are now enrolling patients.
Bemdaneprocel uses iPSC-derived dopaminergic precursors. It targets Parkinson's disease. As of 2025, it is in Phase 3 clinical trials.
Presenting the Fertilo clinical program. It uses iPSC-derived ovarian support cells. The indication is for oocyte maturation. As of 2025, it enters OPCT-001 utilizes iPSC-derived RPE cells. It targets retinal degeneration. Currently in Phase Y2A Clarico trial. Our clinical program focuses on neural progenitors. We use IP indications are ALS and spinal cord injury. As of 2025, we have granted IND clearance. These models mimic early human life, blurring the lines of bioethics. Where does potentiality begin? In April 2025, the ISSCR issued updated guidelines. They address stem cell-based embryo models. Key rules prohibit ectogenesis and mandate strict ethical oversight. These models must never be transplanted. They may acquire moral status, necessitating a shift in governance. Stem cell biology has matured into a therapeutic reality. Classification of cells allows for targeted application, from oncology to regenerative medicine. Advanced purification techniques now mitigate teratoma risk. Expedited pathways pave the way for off-the-shelf products. The boundary between discovery and clinical implementation dissolves.
Like what you share.
This website makes learning so easy. I found all my resources here.
Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.